Welcome to NanoTemper's webinar on biologics characterization. Glad you all could join us. My colleague Charles will present and questions will be taken at the end. So allow me to introduce our speaker, Charles Heffern. Charles received his bachelor's degree at the University of Southern California before doing his graduate studies at the University of Chicago. He's been with NanoTemper since 2015 and is currently the product manager for the Prometheus system. He's a very sharp and personable guy, and I'm going to pass the presenting rights to him to get us started.
Thank you, Andrew, for the kind introduction, and hello, everyone. I do appreciate you taking the time today to learn how the Prometheus can help improve and accelerate your biologic characterization, and in this crowded space is proving to be the new gold standard of biologic stability characterization. So if you don't mind, I'm going to go ahead and get my laser pointer up before we proceed, and there we go. Okay, and here we are. So I'd like to start off this presentation with a little background on the field and why this topic of biologic stability characterization is so important. As I'm sure most of you know, biologics are increasingly important to the treatment of a wide variety of indications and diseases, and year over year, more and more biologics are entering the clinic and receiving regulatory approval from both the FDA and EMA. Not only are these biologics important to the patients whose lives they help, biologics are also increasingly important to the companies that develop them. The effectiveness of these molecules at treating some of the most important diseases has resulted in them becoming the top revenue-earning pharmaceuticals. This combination of therapeutic effectiveness and revenue potential has, of course, spurred increasing investment in a healthy pipeline of clinical candidates. However, there are significant pressures on the development of biologics related to the time and cost of development and the need to increase the efficiency of this process. Biologic development is a resource-intensive endeavor, which is only becoming more expensive and difficult as the structural complexity of these molecules increases. This results in greater difficulties to receive regulatory approval, all while markets are demanding greater efficiency and development to keep prices constrained. To get a biologic approved by the regulatory bodies, either the FDA or EMA, both functionality and stability of the candidate molecule needs to be shown. Historically, biologic development heavily favored the functionality or activity of a candidate molecule, and stability was viewed as a property that could be engineered or formulated in. However, as the increasing development costs and time show, this is not always feasible. A more modern and balanced approach to biologic development is to consider the activity and stability of candidate molecules as equally important molecular properties that must be understood and characterized at all stages of discovery and development to ensure a successful development process. Here, we are talking about the entire life cycle of the biologic, starting from early stage discovery with the developability assessment of all candidate molecules, all the way through pre-formulation and formulation activities, into the process development or production optimization, and the entire clinical life of the biologic, with comparability assessments to address any modifications to production processes or locations. When talking about biologic stability, one of the simultaneously wonderful and frustrating things is the complexity of what stability means. With the complex structure of biologics, stability becomes a complicated matrix of multiple molecular properties, with failure of any one potentially dooming the successful development of a candidate molecule. These properties include the self-interaction and cross-interaction of the biologic with itself or other molecules, its hydrophobicity, its thermal stability or thermodynamic stability, I skipped one, its colloidal stability or propensity to aggregate, and its chemical stability or susceptibility to chemical modifications such as loss of amino acid side chains. This list is by no means exhaustive, and a robust body of literature exists exploring all the different ways the stability of biologics can be characterized and understood. With such a complex matrix, it is critically important that the answers for each property that comprise the biologic stability are clear and lead to actionable insights. It does a research group no good if their attempt to characterize any one of these properties leads to unclear results that just muddy the water. And this is where the Prometheus comes into the conversation as the new gold standard for thermal stability and colloidal stability characterization, providing the most clear and precise answers for actionable insights in biologic development. Here, I'd like to quickly highlight a statement from one of our customers at NGM Biopharmaceuticals, Alan Kutaj, and how the Prometheus enabled him to identify liabilities in his biologic pipeline and invest time and resources in the projects most likely to succeed. According to Alan, his group makes use of the Prometheus to evaluate biologics at early stages and takes advantage of its sensitivity to avoid the costly scale-up of candidate molecules that would be destined for failure. Using the Prometheus, Alan and his group were able to identify an unanticipated structural liability that rendered a molecule unmanufacturable, despite having a robust activity and functional assays. Having identified this liability, NGM was able to focus its resources on more promising molecules and projects. This then brings us to what companies like NGM and scientists like Alan look for when selecting instruments to characterize biologic stability, and what has set the Prometheus apart as the new gold standard. In brief, an instrument for biologic stability needs to be able to provide a high level of specificity or the ability to distinguish the signal of the biologic from the buffer or matrix, a broad limit of detection, or the ability to measure samples at clinically and developmentally relevant concentrations, including both high and low concentrations, a high level of sensitivity, or the ability to identify and measure samples and domains with subtle signals, a high level of accuracy, or the ability to report true values, a high level of precision, which is also the ability to provide consistent and reliable results with small standard deviations or errors that do not vary from day to day or across different positions within the instrument. And finally, a high level of resolution, or the ability to distinguish domains with similar values of stability. At this point, I'd like to turn to a case study from an article published in Analytical Biochemistry by a group working at Amgen on comparability studies of biologics and therapeutic proteins, and how they showed that the specificity, sensitivity, accuracy, precision, resolution, and limit of detection of the Prometheus made a difference in their biologic characterization where other instruments struggled. Moreover, one thing that I personally like about this case is its focus on comparability studies, which are a regulated process overseen by the regulatory bodies to validate that changes to manufacturing processes or sites do not affect the biologic drug product. Starting off with the specificity and accuracy of the Prometheus, the group at Amgen benchmarked the Prometheus to the traditional standard for thermal stability characterization, differential scanning calorimetry. In their results, the group showed that the Prometheus was at least equal to the traditional standard on 1, specificity, as highlighted in the left figure, with no signal from the formulation buffer as shown in the black trace, compared to a clear signal from their pipeline bispecific T-cell engager, or BiTE molecule 1, shown in blue. And 2, accuracy, as highlighted in the right figure, where with the Prometheus data shown in blue trace contrasted to their DSC in the black trace. Moving on to the precision, resolution, and limit of detection, it was shown that the Prometheus surpasses the traditional standards and is unmatched. As shown by the authors of this case study, the precision, or as they name it in their article, the repeatability of the Prometheus is within 1/10 of a degree Celsius for all molecules, with 7 shown in this figure, lots, with 3 different lots of the same biologic shown here, and concentrations thrown at it. I'd like to pause for a moment to have a closer look at the consistency of the precision shown in this figure across a broad range of concentrations, ranging from tens of micrograms per mil all the way up to almost 100 mg per mil. This unmatched precision holds true for all antibody domains with the Fab, CH2, and CH3 shown here, along with different biologic constructs such as bispecifics shown here. And one thing I would like to quickly highlight about the data on the repeatability or precision as shown in this figure is that all of these measurements were performed in septuplicate for a very thorough assessment of that precision. It is this combination of best-in-class properties that make the Prometheus the new gold standard in biologic stability characterization. Even as the limit of detection, sensitivity, or resolution are pushed to the extremes, there is no compromise on the precision or other critical measurement properties. The Prometheus also enables measurements that use less time and sample, that work for a broader range of molecules, as well as across a broader concentration range, and that are fully automatable. I'll get into some of the specifications about these last 5 points as we proceed through the presentation. While the case study from the Analytical Biochemistry article by the group at Amgen did an excellent job highlighting the qualities of the Prometheus, I wanted to quickly bring up some specifications that further explain and define the repeatability and precision of the instrument. This data can be found in a tech note on the precision of the Prometheus. What we are looking at in this figure is an overlay of the thermal unfolding traces of 48 measurements of the thermal stability of streptavidin at 1 mg per mil, all measured simultaneously. It can be a little tricky to see that there is more than one data set shown in that plot, but I promise you, there is data from 48 independent measurements in that figure. The repeatability or precision of this TM was within 1/10 of a degree Celsius, and the consistency or precision of the fluorescence detection, which is used to determine the structure and unfolding of the protein via its fluorescence ratio, to within 8 thousandths. It is this precision of the fluorescence detection that results in the unmatched sensitivity of the measurements on the Prometheus. For groups that require consistency of results across multiple groups or sites, or are working with collaborators or contractors such as a CRO and require consistent results, it is the intermediate precision that shows the consistency that you can and should expect. These values are generated by comparing the results of 10 instruments on each of which 48 samples were measured, testing all positions within all 10 instruments. Even switching between 10 different instruments, the repeatability or precision of the TM was within 0.3 of a degree Celsius, and the consistency or precision of the fluorescence ratio was within 0.02. Here, I'd like to highlight a statement from another scientist who uses the Prometheus to push the limits of biologic characterization, throwing complicated proteins and formulations at the Prometheus, yet continues to obtain high-quality, reproducible results that help Donnie and BioMarin identify the projects that are going to fail early and eliminate them from their pipeline so they can focus their efforts on the molecules that have the most promise in the clinic and get the high-quality molecules that will make a difference to patients into clinic faster. In addition to the thermal stability measurements featured so far, the specificity, sensitivity, limit of detection, accuracy, precision, and resolution of the Prometheus can also be applied to a number of additional stability indicating properties of biologics, including chemical unfolding, delta-delta G measurements, isothermal and thermal cycling measurements, and of course, antigen buffer and assay optimization for the complex therapeutic targets that comprise the high-hanging fruit targeted in the development of many modern biologics. At this point, I'd like to turn our attention to another case study. This study, published in the journal Scientific Reports by a group working at Janssen, shows how the specificity, sensitivity, accuracy, precision, resolution, and limit of detection of the Prometheus, along with its versatility to both thermal stability and chemical denaturation measurements, facilitated the engineering of stability enhancements in an antibody derived from a transgenic animal platform. Moreover, the group goes on to highlight how these results relate to optimization of the behavior of the antibody in storage and accelerated degradation assays and an optimized immunogenicity profile. When characterizing and developing this antibody, called PCA75, derived from a transgenic animal platform, the group, led by Sathya Venkataramani, identified several rare somatic hypermutations that could potentially lead to a compromised immunogenicity profile. The mutations, highlighted in a structure sequence alignment, include arginine at position 14, which is here, proline at position 20, histidine at position 81 of the heavy chain, and alanine at position 1 of the light chain. The relative positional frequency of the amino acids in each of these positions can be seen on the right in the relative frequency plot. Now, I'm not going to profess to be an expert on the development of rare somatic hypermutations in antibodies developed in transgenic animal platforms, but what I can and will address is the work performed by this group at Janssen to assess the effect of these mutations on the stability and immunogenicity profile of this antibody. Through a systematic introduction of the most common amino acids found in positions 14, 20, and 81 of the heavy chain, and position 1 of the light chain of the human IgG4 germline, the group at Janssen leveraged the capabilities of the Prometheus to probe the effect of each substitution on the thermal stability and aggregation onset of the monoclonal antibody, focusing on the effects on the Fab, while validating that there was no loss in the activity or functionality of the candidate molecule using binding assays. Through their work, they found that the introduction of the most common amino acids at each of the positions of the somatic hypermutations resulted in an increase of FAB-TM of 8 degrees and TAG of 14 degrees relative to the parent PCA75 antibody, as seen with the variant PCA62, shown here in bold. Additionally, the accuracy and precision of the Prometheus was again validated through a thorough comparison of thermal stability measurements using both the Prometheus and differential scanning calorimetry. Speaking of thorough, the versatility of the Prometheus was leveraged by Sathya and her group to validate the effects of the point mutants on both full-length antibodies, PCA75, and the optimized engineered version, PCA62, as well as the effects on the Fab domains. With the Prometheus enabling offline incubation of samples for chemical denaturation measurements, the samples were incubated with a chemical denaturant for a minimum of 12 to 16 hours prior to measuring the conformational change to allow ample time for equilibrium to be established to assess the true unfolding energetics without tying up the Prometheus. As determined from the chemical denaturation measurements, the engineering of the Fab resulted in an increase in the Gibbs free energy of unfolding from 34.8 to 42.5 kilojoules per mole, as well as the C50, or chemical denaturant concentration at which half of the antibody is unfolded, from 1.7 molar to 3.6 molar guanidine chloride. Turning to the storage and accelerated stability measurements performed on the parental and engineered monoclonal antibodies, the correlation between the increased thermal stability and chemical denaturation stability of the engineered monoclonal antibody PCA62 relative to the parental monoclonal antibody PCA75 and decreased propensity to form aggregates as determined with analytical size exclusion chromatography, is clear. Here, the formation of aggregates of PCA62 and PCA75 at 4 degrees Celsius over a 4-week span can be seen. And over here is the formation of aggregates of PCA62 and PCA75 at 40 degrees Celsius over a 4-week span, with again drastically reduced formation of aggregates and higher order structures in the engineered optimized monoclonal antibody. While the previous case study showed the power and versatility of the Prometheus for biologic thermal stability and chemical denaturation stability measurements, and correlation of these properties to storage and accelerated stability assays, one of the limitations identified by the authors of the article was that the analysis in the scientific reports article was indeed just a single case study and not an extensive sample set. Here, the Prometheus is again prepared to support the needs of biologic stability characterization with the possibility to operate as either a walk-up instrument or a fully automated system for higher throughput characterization of those more extensive sample sets. The automation is a modular addition to the Prometheus, and the fully automated system retains all of the instrument properties that make the Prometheus the gold standard that it is. In our third and final case study, drawn from an article published in the journal MABS by a group working at Merck, the topic of comprehensive and thorough characterization of an extensive sample set is taken head-on. By leveraging the throughput and ease of use of the Prometheus, along with other instruments to integrate comprehensive stability characterization early in the biologic candidate selection process, Mark's group is optimizing their chances of selecting candidate molecules that can survive the rigors of drug product development. Additionally, we'll have a quick look at correlations between the biologic stability properties as identified in this comprehensive dataset and how a combinatorial analysis of the complex matrix that define a biologic stability using clear, precise results like those provided by the Prometheus helps identify lead candidate molecules with the fewest liabilities and most favorable stability profiles. Starting off with an overview of the characterization workflow, we can see this group at Merck integrates a comprehensive, high-throughput characterization of their biologics from early-stage discovery that is used to inform the molecular design and development decisions. By leveraging the throughput and low sample consumption of the Prometheus, thermal stability and colloidal stability analyses can be conducted from the very early stage using only minimal amounts of sample while still achieving the highest quality results, as seen in the Amgen case study. Having generated such a glut of data, robust and reliable correlations between the various stability indicating parameters can be identified. Here, we see positive correlations in red and negative correlations in blue, with some of the strongest positive correlations for selected analytics tabulated here on the right. As can be seen, in addition to the strong self-correlations between Tag, Tm, and T-onset as determined by the Prometheus, there is also a very strong correlation to the capillary isoelectric focusing and UPSEC main peak. When implemented in a combinatorial analysis of the multiple properties that comprise a biologic stability, as discussed at the beginning of this conversation, the high-quality results provided by the Prometheus provide even more insight on which molecules have the stability properties necessary to be a successful clinical candidate. Looking at cluster 3 from this cluster analysis, we can see that these molecules have the greatest percent monomer according to UPSEC, are the most thermally stable according to the Prometheus, have the fewest self-interactions according to affinity capture self-interaction nanoparticle spectroscopy, which is a mouthful now that I say it, so I see why we write AC-SINs, develop the fewest high molecular weight aggregates at a low pH hold, and are the most basic with a pH away from physiological. Taking a break from the case studies, we spent most of our time so far discussing what is needed from an instrument for biologic stability characterization, how the Prometheus sets itself apart from other instruments, and how groups are using the Prometheus to take their biologic characterization to the next level through the case studies and customer statements. However, we haven't really addressed how the Prometheus actually works, what makes it different from other instruments, and how it is able to achieve its specificity, sensitivity, limit of detection, accuracy, precision, and resolution, not to forget its throughput, low sample consumption, and ease of use. I would like to now take a few minutes at the end of this presentation to address precisely this. Starting off with the basics, the Prometheus is a label-free platform that detects changes in a biologic or protein's intrinsic fluorescence to determine whether it is folded, partially denatured, or unfolded. When a physical stress, such as temperature or a chemical denaturant, causes a biologic or protein to unfold, this change in structure results in a change to the local environment of the aromatic amino acids tryptophan and tyrosine, as they typically become more solvent accessible. In addition to a decrease in the quantum yield of these two fluorescent amino acids, this change in hydration also results in a change to the maximum fluorescence emission wavelength, which typically undergoes a redshift, as seen here. It is these two phenomena that are measured by the Prometheus to detect the stability of biologics and unfolding caused by either thermal stress or a chemical denaturant. Now, there are many instruments that can measure similar phenomena, so what really sets the Prometheus apart is the way the instrument and optics are engineered to measure this fluorescence with the greatest sensitivity and precision. While most other instruments make use of full spectrum detectors, the Prometheus is designed around a set of highly sensitive and specialized optics, lenses, and photomultiplier tubes to achieve this unmatched level of performance. Simultaneous to the nano-DSF measurement, the Prometheus also detects the formation of aggregates using our back reflection technology by passing light in the near UV through the samples. In the absence of aggregates, there is 100% transmission of the near UV light. As aggregates are formed, the near UV light is scattered and the transmission decreases proportionally. This facilitates detecting not only the onset of aggregation, but also the size and extent of aggregation. It is the combination of these two seemingly simple measurement technologies in a single high-quality instrument designed to record the highest possible quality data and provide clear and unambiguous results that facilitates the research highlighted in the case studies and described by the customer statements. To speak quickly to the workflow, samples are loaded into capillaries that can either be loaded individually or with samples, or directly from standard 384 well or 96 well plates. These sample holders can be held and manipulated by robotic arms for full walkaway automation. A thermal ramp can be defined anywhere from 1/10 of a degree Celsius per minute all the way up to 7 degrees Celsius per minute, all of which can be performed with no loss to data quality or resolution thanks to the design of the Prometheus, with thermal ramps ranging from 15 to 95 degrees Celsius and can be extended up to 110 degrees Celsius when required. Fluorescence excitation is performed with a precise LED source. Just under 10 microliters of sample is required per capillary, and samples can be measured at concentrations ranging from as low as a few micrograms per milliliter, all the way up to hundreds of milligrams per milliliter. And what's really kind of fun and interesting to me personally is that both extremes have been shown to be necessary and valuable for the characterization of biologics, depending upon the potency, quantity produced, and desired mode of administration for the final drug product in the clinic. With that, I would like to thank you for your time and attention and pass this back over to Andrew and open things up for any questions that you may have.